Dear students, welcome to EPG Pathshala. The module title is Completely Randomized Design 1 is in continuation of the previous module, that is Basic Principles of Design of Experiment. In this module, we will discuss what is ANOVA, that is Analysis of Variation. As we already know that for the testing of mean we have t-test, but that we can apply only in the situation when we have only two samples. For more than two samples and for the testing of mean of the samples, we use ANOVA test. In this module we will discuss analysis of variance. After that we will discuss that fixed effect models that we will use in this module. After that we will discuss how we can split total variation into different components, and after that we will discuss estimation of the parameters. These are the most important thing. Then one should know that how to estimate the parameters for the models, and at the last some examples are here for better understanding. Hope you enjoy it. Let's get started. Our main objective is to give an introduction to completely randomized design, or ANOVA. The two effects of completely randomized design model, first is fixed effect and random effect models, will be discussed in detail. Statistical analysis of the model as well as the mathematical deviation for the construction of ANOVA table will be discussed. This model has played a vital role in different areas of research. Example will be discussed to understand the topic in depth. In general, the experimenter is interested to investigate whether the observed difference between the treatment mean are non-significant, that means it occurs by chance, or whether they exist an actual difference between the treatment means. With the help of experimental design that enables to compare the effect of different treatment on the response variable mean value. In many situations, it is possible that the effect of treatments on the mean of the response variable to be interested is only due to the variability that occur from the extraneous factor that are not of interest. To take care of this situation, a proper experimental design is chosen to get an appropriate results. We already discussed about three principles, that is randomization, replication and blocking in the module Basic Principle of Design of Experiment. These three principles will help the experimenter to concentrate on the objective response variable and the statistical analysis as well as the estimation of the treatment effect, and how to take care of the extraneous, uncontrolled and non-assignable factors while building a model. Basically, the completely randomized design is used to split the total variability into two segments. It follows the method of separating the total variability into components that have different source of variability. These sources of variability are then compared, and this comparison will help to test about the significance of the null hypothesis that there is no difference exists between the treatment mean. It is essential that one should have a clear idea about testing hypothesis and statistical terms involved in them. One can refer to the modules based on the testing of hypothesis and some parametric test. The test of completely randomized design is very important as it has wide application in all the areas. To make this module simple and understandable, we split this topic into two modules. In this module, we will discuss about the theoretical part of ANOVA, including assumptions of the model, decomposition of the total sum of squares, estimation of the model parameters, and statistical analysis. In the next module, we will discuss in brief about checking for the assumption of the model and the pairwise comparison of the treatment with various tests available in the literature. First, we will discuss that what is ANOVA, that is analysis of variance. In this section, we will discuss about the analysis of variance of a single factor experimental design problem. This single factor design contains one types of factor that affect the result of the experimental design. This single factor design is also known as completely randomized design. ANOVA is the abbreviation for the one-way analysis of variance. Suppose there are P treatments or different levels of single factor. Our objective is to compare P treatments or different levels of a single factor. Observations are recorded with respect to each of P treatments, and all observations are treated as realization of random variables. So we have a table that contains the values of the variables, where y11 denote the first observation from the first group. That means that we have y1 denotes the first observation from the first treatment, and similarly we have yp1, that is the first observation from the pth treatment, and so on. And totals of these row sum is given here as y1 dot, y2 dot, and yp dot, so and so on. So, and y double dot represent the grand total, and y1 dot bar represents the average value of the first treatment, and similarly yp dot represents the average value for pth treatment. We can see that observation of the response variables are represented in terms of y, ij, where I and j vary from 1 to p, is used for treatments or different levels of a single factor, and j vary from 1 to n, represents the observation with respect to ith treatment. We can see that there are p treatments and n observation in each treatment, hence the total number of observations are np. Some of the notations of ANOVA, that y11, that is first observation with respect to first level of the single factor, and y1n, nth observation with respect to first level of the single factor, ypn, nth observation with respect to the pth level of the single factor, and y1 dot, that is total of the all observation with respect to first level of the single factor, and yp dot, total of the all observation with respect to pth level of the single factor, and y double dot represent the grand mean, that means grand total of all the observation with respect to all levels of the treatments, and yp dot bar, that is mean of all the observation with respect to pth level of the single factor, and y dot dot bar, that is grand mean of all the observation with respect to all levels of the treatments. Now, what is a statistical model? The statistical model is given here as, for that we have to understand the terms, that is what is parameters and what is error term. So we should have to build a model for the recorded single factor observation with different levels. The model is similar as the one-way ANOVA statistical model. The model is yij is equal to mu I plus error ij or epsilon ij, where I takes vary from 1 to p and j vary from 1 to n, and yij, that is ijth observation in the design, and mu I is the mean of the ith level of the factor, and epsilon ij the error random variable that has zero mean. So error terms are independently distributed random variable that follow normal 0 sigma square. Under this model, the error terms are uncorrelated with the treatment, so yij are independent and it is distributed as normal mu plus tau I comma sigma square. So if we want to write another form, then we have to put the value of mu I as mu plus tau I. So if we put this form in the original model, then the modified form of the model is yij is equal to mu plus tau I plus epsilon ij, where mu is the overall mean, tau I is the ith treatment effect. The original model is also called the mean model, and it involves the effect of the different treatments or levels of single factor due to its mean value. Now after transforming the original model, the new model is known as effects model. Both models are linear statistical models, and also because both are very useful in the experimental design literature, but effects model is widely used because it seems to be more appropriate to incorporate constant mean and treatment effect separate in the model. In the treatment effect model, we can see two types of effect. One effect if the treatments are chosen by the experimenter. In this situation, we are interested in to test about the treatment mean, and after applying the statistical analysis, the conclusion about the significance of the hypothesis, that is whether to reject the null hypothesis and do not reject the null hypothesis. And this is only applicable for the treatment taken in the study. The result cannot be generalized to other treatment that were not considered. Hence, this type of effect is known as fixed effect model. Second effect is when the treatment would be taken as a random sample from a large population of the treatment. The conclusion by the testing of hypothesis may apply not only for this random sample but also to other random sample treatments of the population. This effect is known as random effect model. This model is also known as components of variance model because in the model treatment effects are random variable, and we try to test about the variability of the treatment effect and also try to estimate the variability among them. Now the theoretical part of analysis of fixed effect model. In this section we will discuss analysis of variance for the fixed effect model. Our objective for this analysis is to compare the mean effect with respect to different levels of the single factors. Let yi dot bar be the average of the observation under the ith treatment. Similarly, y double dot be the total of all the observations, and y double dot bar denote the total average of all the observations. The mathematical form are given as yi dot is equal to j yij, where summation is over j and j vary from 1 to n, and yi dot bar is equal to yi dot divided by n, where I vary from 1 to p, and y double dot is equal to yij and summation is over I and j, where I vary from 1 to p and j vary from 1 to n, and y double dot bar is equal to y double dot upon capital N, where capital N is equal to small n into p, where it represents the total number of observations. So in the fixed effect model, we are interested in the testing the hypothesis of the equality of treatment mean. The hypotheses are given here as h naught, which is equal to mu1 is equal to mu2 is equal to mu p, which is equal to mu. That is our null hypothesis. So our alternative hypothesis is where mu I is not equal to mu j for at least one pair of I and j, as mu I is equal to mu plus tau I, and summation over tau I, where summation is over I and I vary from 1 to p, is equal to 0. Then the hypothesis in terms of tau is defined as h naught, tau 1 is equal to tau 2 is equal to tau p is equal to 0, and alternative is tau I is not equal to 0 for at least one I. Some of the assumptions of ANOVA are given here as there are certain assumptions for the analysis of variance. These are the population must be from normal distribution. Second assumption is the population must have a common variance, and third is the observation must be independent. One has to test these assumptions before applying the model on the data. To test the assumption of normality and independence of observation, one has to be familiar with the method available in the literature for this purpose. Now we will discuss about how we can separate the total variability into components. So in this section, how we can split the total variability into component associated with different sources of that variability. The total sum of square is defined as sum SST, which represents sum of square of treatment, which is total, which is equal to double summation y ij is equal to y double dot bar, and summation is over I and j. The above expression can be written as the double summation yij minus y double dot bar whole square, which is equal to double summation yi dot bar minus y double dot bar plus yij minus yi dot bar whole square. The above expression can be expanded further. The final form that we will get is given here as double summation yij minus y double dot bar whole square, which is equal to n times summation yi dot bar minus y double dot bar whole square plus double summation yij minus yi dot bar whole square. Hence, the total variation in the data can be segmented into sum of square that we derived by subtracting the total average from the treatment average plus the sum of square of difference of the observations from the treatment mean. The symbolic notation for the above expression is given as sum of square of total, which is equal to sum of square of treatment plus sum of square of error, where sum of square of treatment is called the sum of square between treatments and sum of square of error is called the sum of square due to error term. There are capital N observations in total, thus sum of square of total has n-1 degree of freedom. There are p levels of the factors, so sum of square of treatment has p-1 degree of freedom. So the remaining term, that is error, has n-p degree of freedom. Now the next section is how we can estimate the parameters, and it is very essential that you must know that how the parameters are estimated in the model. Now we will discuss about how to estimate the parameters of the model. The parameters are mu and tau I, where I vary from 1 to p, are estimated by using the method of least square theory. The error term is noted as Eij and it is defined as Yij minus mu minus tau I. The error sum of square is given here as represented by a capital letter Q, and which is equal to double summation Eij, and summation is over I and j, where I vary from 1 to p and j vary from 1 to n, and which is equal to double summation Yij minus mu minus tau I whole square, and summation is over I and j, where I vary from 1 to p and j vary from 1 to n. We want to estimate the parameters, that is mu and tau I, in such a manner that our error term should be minimized. For that, differentiate the function q with respect to the parameter mu and tau I and equate them to 0. After differentiating the function q with respect to mu, we get the term as minus 2 times double summation Yij minus mu minus tau I, and after differentiating the function q with respect to tau I, the expression we will get is minus 2 times I vary from 1 to p and j vary from 1 to n, Yij minus mu minus tau I, where I vary from 1 to p. After equating to 0, we will get the following equation y double dot, which is equal to np mu plus n times summation is over tau I. Also I vary from 1 to p, tau I is equal to 0. So the estimator of the mu is mu hat is equal to y double dot upon np. Hence the estimator of mu is y bar double dot, which is the overall mean of the observed responses. Similarly, estimator for tau I are given as tau I hat, which is equal to yi dot bar minus y double dot bar, where yi dot bar is the mean of the ith response. After estimating the parameters of the model, now the next step is to estimate the parameter sigma square for the ith treatment response. So which is, as we know that the SI square is an unbiased estimator of sigma square. So 1 upon sigma square summation of Yij minus yi dot bar whole square follow chi-square distribution with n-1 degree of freedom, as S square follow chi-square distribution, and this is the property that if any variable that follow normal distribution is some parameter, if you scale it, then it follow chi-square distribution with 1 degree of freedom. So use the above information, we can find an estimator of sigma square. So as we know that for the property of unbiasedness, if you take expectation of the estimator, then if it is coming out to be equal to the parameter, then we can say that parameter is estimated by this particular function. So that particular function is called an estimator. So here we are taking expectation of summation of n-1 SI square, which is coming out to be n-p into sigma square. So sigma hat square, which is equal to 1 upon n-p summation n-1 SI square, so which is the pooled estimator of sigma square. So 1 upon sigma square double summation Yij minus yi dot bar whole square follows chi-square with n-p degree of freedom. From the section double summation Yij minus yi dot bar is represented by sum of square of error, and so the estimator for the sigma square is sigma square hat, which is equal to sum of square of error divided by n-p. Hence sum of square of error divided by sigma square follow chi-square with n-p degree of freedom. As we have defined the null hypothesis as the treatment mean are equal to the treatment effect or 0, So here h naught, which is tau I, is equal to 0 where I vary from 1 to p, and h1 is tau I not equal to 0 for at least one I. When h naught is true, SST, that is sum of square of total divided by n minus 1, is the overall sample variance, and SST upon sigma square follow chi-square with n minus 1 degree of freedom. Now in this section we discuss about the important theorem, that is Cochran theorem. It is essential from the point of view for understanding the F statistics, that how we apply this theorem for the construction of the ANOVA table. So this theorem states that let Zi follow normal distribution for all I vary from 1 to p, then summation over Zi where I vary from 1 to p, which is equal to W1 plus W2 is equal to WA, where A is less than equal to p and Wi has pi degree of freedom where I vary from 1 to a, then W1, W2, Wa are independently chi-square distributed random variable with P1, P2, Pa degree of freedom respectively if summation, if P1 P is equal to P1 plus P2 and so on Pa. Now in this section we discuss how to construct ANOVA table. For that we use Cochran theorem, and from the Cochran theorem we can see that sum of square of error and sum of square of treatments are independent. It follows that sum of square of treatment divided by sigma square is chi-square with p minus 1 degree of freedom, and also sum of square of treatment with p minus 1 is another unbiased estimator of sigma square. So now we also know that proportion of two chi-square variables follow F distribution to test whether the treatment variance is significant or not. For this we take the ratio of two chi-square variable with respect to their degree of freedom. So that is equal to mean sum of square of between treatment divided by mean sum of square of errors that follow F distribution with p minus 1 n minus p degree of freedom. And h naught is rejected for large value of this proportion. To make a decision about the hypothesis, we compare the value of this ratio with the tabulatory value from the table. The test score is one-sided test. From the ANOVA table we can see that on the in the first column we have source of variation, and the first source of variation is between treatment, second is residual, and third one is total. And in the second column we have degree of freedom corresponding to the source of variation, and in the third column we have sum of squares, and in the fourth column we have mean square, and then the fourth column we have F statistics. From the above table we can see that for calculating F statistics one has to derive sum of square of treatment, sum of square of error first. Hence it is essential that one must be able to calculate these sum of squares and then calculate the F statistics. This is an example that will help you understand how we can apply the ANOVA table in our practical life. A random sample of marks out of 50 students is in the mid-semester examination in statistics from environmental studies, biostatistics and human genetics department from Central University Punjab were taken respectively. The results were as follows, and we have observations available for the experimental studies, biostatistics and human genetics students, and these observation represents the marks of the students. Here y1 dot, that is the sum of the students, sum of the marks of the students from experimental department, and y2 dot for biostatistics department, and y3 dot represents the sum of the marks of students from the human genetics department. And n1 here is 10, n2 is equal to 8, and n3 is equal to 9, and double summation yij square which is equal to 3052. The formulation from sum of square of total is equal to double summation yij whole square minus y double dot square upon n, where n takes value n1 plus n2 plus n3. So after putting the value you will get the result as sum of square of total, which is coming out to be 1470.52. So also the formula for the sum of square of treatment, which is equal to summation yi dot square upon ni minus y double dot square upon n. Hence sum of square of treatment, which is equal to after putting the sum and square them divided by the number of observation, we get the result at 356.09. Also the sum of square for the error can be found by subtracting using the relationship, that is sum of square of error which is equal to sum of square of total minus sum of square of treatment. Therefore sum of square of error which is equal to 1470.52 minus 356.09, which is coming up to be 1114.43. So the solution is represented in table number 3. So as we know that we have 3 groups, so the degree of freedom for the between department is coming to be 2, and source of variation after putting the value in the formula, and we get the results as given here as, and the f statistics value is coming out to be 3.83. so hence we have a null hypothesis that h naught, which is equal to tau 1 is equal to tau 2 is equal to tau 3 is equal to 0. The tabulated value of the f statistics with degree of freedom 2, 24 at 5% level of significance is taken from the following table, figure 1, that is given in the appendix 1. So we can see that the value is coming out to be 3.403. As the calculated value of the f statistics is greater than the tabulated value, that is f with degree of freedom 2, 24, hence we reject the null hypothesis at 5% level of significance. It means that there is a significant difference in the marks obtained by students from different departments. Now we will discuss another example, and in this example we have 2020 match Indian team score, different runs scored by different teams and against different country teams. In a total of 15 games, Indian team score total run as given in the following table. So on the left hand side we have some teams. That first team is Sri Lanka, second Australia, third is South Africa, and the score scored by Indian team are given in the second column. From the data, first we have to find out the unbiased estimate of the overall mean, and second to find out the unbiased estimate of the treatment effect for each treatment, that is tau I. Then construct an ANOVA table to test whether there is any significant difference between the runs scored by Indian team against different country teams at 5% level of significance. And fourth is, what are the assumptions for the model? From the available data, first calculate the sum of runs scored by Indian teams against each country team and the total sum scored by Indian team in total 15 matches. So y1 dot is equal to 900. That means the score scored by the Indian team against Sri Lanka is coming out to be 900 total score, that means, and y2 dot is the total scored by the Indian team against Australia team, that is coming out to be 1215 runs, and similarly y3 dot is equal to 684, and y double dot, that means the grand total scored by Indian teams against different country teams, that is 2799. To find an unbiased estimator of the overall mean, so we know that the unbiased estimator of mu hat is y double dot bar. So sum all the total values and divided by the total matches, which is coming out to be 186.8. and the second estimate for the treatment effects are, so it is the formula for estimator of treatment effects is given as y1 dot bar. So after putting the value in the formula, you will get the result as 180, and tau 1 hat is which is coming out to be 180 minus 186.6, and which is equal to minus 6.6. Similarly, values for the another parameters are given here as 15.9 and minus 15.6. The null hypothesis is given here as h naught, which is equal to tau I is equal to 0, where I vary from 1 to 3, and h1 is tau I not equal to 0 for at least one I. For calculating the f statistics, we have to derive the sum of square of total variability, sum of square of treatment. Sum of square of error can be calculated by subtracting sum of square of treatment from the sum of square of total. So it is coming out to be 526901. Hence put the value in the sum of square of total, we get sum of square value is coming out to be 4607.6. To calculate the sum of square of treatment, we have the formula that we have to take the sum of score of Indian team scored against Australia and Sri Lanka and other team. We have to take the square of the sum and divided by the matches that is played by the Indian team against them. So it is coming out to be 2708.1. Now calculate the sum of square of error, which is which is coming out to be 4607.6 minus 2708.1, and it is coming out to be 1899.5. So the from the table we have, after solving different sum of square, we put the value in the table here, as the calculated f statistics value is 8.554. Now from the figure 1 given in the appendix 1, that is given in the e-text at the end of e-text, so at 5% level of significance, calculate the value of f2, 12. It is 3.885. The value of test statistics is very high than the tabulatory value. In other words, calculated value of f is greater than f with degree of freedom 2, 12. So we reject the null hypothesis, which states on average basis runs scored by the Indian team is same for all country team. So we can say that there is a strong evidence from the data given above that the runs scored by Indian teams is slightly different from different country teams. The result that we have derived are based on some assumptions. The assumption for this problem is that runs should conform to normal distribution and variance should be same for all the teams. In the end, I would like to conclude this module as given here as in this module I discussed that what is ANOVA, that is analysis of variance. In this module we discussed that how we apply ANOVA in our practical life, and for this we also discuss in this module as that is what is fixed effect model and random effect model, the difference between two of them. And after that we also discussed that how we can split the total variation into different components, that is between treatments, between sum of square and so on. We also discussed that how we can estimate the parameters of the models, and after estimating the parameters of the model, how we can find other values in this module. Thank you.